So just how do viruses and other nasty things get into our cells? Don't our cells have a membrane, something to protect the cell? Haven't we evolved these highly selective membranes to keep things out and let some things come in? In this video, we're going to talk about just how things go into and out of the cell, including those nasty viruses. Let's get started. So your cell does have a protective barrier. We call that a cell membrane, or some people call it a plasma membrane, because we need to have a cell that keeps some things inside and some things outside. So the cell membrane is just a natural boundary. So here you can see our cell membrane. Now let's think about this. Things do need to go in and out of a cell. So what are some things that might go into a cell? Well, of course, right? Food. You eat food, your cells need that food to process and make energy. So you need carbs, right? These smaller sugars, proteins, amino acids, lipids, water, salt, all of these major macromolecules that we've already talked about, right? That your body is made of and your cells are made of. All these major macromolecules, those need to go in. Okay, well, what needs to come out? Obviously, things like waste. Okay, ammonia, some salts, carbon dioxide, another byproduct. Some water occasionally needs to go out. So things need to go in, some things need to go out. How is that accomplished? Well, it has to be accomplished through this plasma membrane. We need something that is semi-selective or semi-permeable. Some things can go in, some things won't go in. All right, so if we're going to make a cell membrane, or if you were designing a cell membrane, what of the four major molecules would you use? Well, if you remember, we talked about the idea that lipids are a perfect molecule to make up cell membranes because when they combine with a phosphate, they make a phospholipid, which has two sides, hydrophobic and hydrophilic. So let's look at a phospholipid. So here we go. There's a typical phospholipid. Here's the chemical structure, as you can see, right? We've got a phosphate group up here at the top, and then we've got these fatty acid chains here. Remember, this one happens to be unsaturated. That one's a saturated fatty acid. So if you're looking here, you'll see, all right, so we've got a head that's got a phosphate group. We've got these tails that are kind of fatty acids. You may remember that the phosphate group is hydrophilic, whereas the lipid tail is hydrophobic. Now, these are going to make great membranes for this very reason. So if you take a bunch of these phospholipids and you get a layer there and you get a layer there, what you'll see is we have a bi-layer, two layers, right? So we've got the hydrophilic heads on the top. Those can be close to water, no problem, right? But the hydrophobic tails in the center, right, they're not going to really allow lots of water to move through there. They're hydrophobic, right? Water doesn't work well there. So we have this interesting situation where these phospholipids make up our membranes, make up this bilayer, because the outside, let's say this is the outside of the cell, can touch the water. The inside of the cell, let's say this is the inside, can touch the water, but other things are not necessarily going to hang out in the center, like that water, because this is of the hydrophobic tails. All right, so phospholipids make up this selectively permeable membrane to keep things in and keep things out. So let's look a little deeper. What kinds of things can go in? What kinds of things can go out across that membrane? So here we have a nice cell, right? Here's the inside of the cell. Here's the outside, the extracellular matrix. So let's think about it. What molecules can go through directly? Well, this layer, right, is made of phospholipids. So things like lipids can pretty easily just pass through. So if we got a lipid right there, it can just go right through. Also, small things, small uncharged polar molecules, As you can see here, a little bit of water in small amounts. A lot won't get through, but little bits of water can get through. And then also small non-polar molecules, oxygen, CO2, N2. And so if you take a look, there's some carbon dioxide that can go right across. We'll also see some oxygen, right? That can diffuse right across. And then, what about other things? Ammonia. We said that ammonia, a waste, needs to go out of the cell. Ammonia can't just go right across that membrane. Take a look at sugar. Oh, we need sugar in the cell, right? We need to make energy. Amino acids, other important things. All of these important things that need to move across the membrane, they can't just slip right through that membrane. So how do they do that? How do they get across the membrane? How do these other things move across? Well, large polar molecules like amino acids and ions, like these charged particles here, will have to move through a membrane protein. So here we got a charged potassium, right, moving right across. Notice that it went right through the membrane. Also notice here that, sodium's going through there. Now notice, sodium and potassium used different proteins. They use different channels to get through. There's not sort of one channel fits all for everything, right? These are specific protein channels for each specific different substance that needs to move across. And so here we have another one. Oh, there's one for particular amino acids, right? Then if we have a large amount of water, it'll move through a channel protein we call an aquaporin. So there's an aquaporin, and there's lots of water that needed to move through. Okay, so lots of things can just go directly across, the things that we talked about in the last slide. But some things, some of these larger molecules and charged ions, things like that, they need to use one of these channel proteins to move across the membrane. So, how do we build this membrane that needs some things to permeate? It's semi-permeable, right? Semi-permeable, some things go across. And of course, we talked about the phospholipid being amphipathic. That's just a fancy word, you remember, for both hydrophobic and hydrophilic. So here we are again. We got our head, we got our tails down here, and then we got this other layer, right? So this is, again, a bilayer. So pretty much, this is going to let a few things through, like we talked about. But we need these other proteins or channels to help us out, because proteins also can be amphipathic, having a hydrophobic and a hydrophilic part. They'll be able to transverse this bilayer. So there's a particular protein. Now, think back to our discussion of proteins. Do you remember that proteins are made of amino acids, right? It's just a long chain of amino acids, right? So here's an amino acid, there's an amino acid, there's a long chain of amino acids. You also remember that they fold. And they fold because of interactions between the amino acids. And some amino acids, you remember, are hydrophobic. They like the water. They don't like the water. Some are hydrophilic. So if you think about that, the protein is going to orient itself in the membrane to have the hydrophilic part out here. It's fine with water. But that hydrophobic part, it doesn't want to be close to the water. So it's going to orient itself here in the fatty acid chains. This will help it anchor and be a great protein and channel that other things can move through. So why these proteins? Well, I just mentioned it. The amino acid chains, right, all have these different R groups. And these R groups, because they're different, give us different properties. The hydrophilic amino acids, as I mentioned, will stick out here in the watery fluid. And these are going to be good for little receptors, right? So when something wants to come in, it might bind to the receptor and then be allowed to come through. The hydrophobic part of the protein, remember, these have the amino acids that are hydrophobic, so they're going to be anchored here in the membrane. So this is going to give it some anchoring, so the thing's not moving in and out so easy. So because proteins have, or could have, both hydrophilic and hydrophobic amino acids, they are great at anchoring across a membrane and allowing other things to come in and out. So let's look kind of at the bigger picture here. So once again, we've got our cell. Here's the outside of the cell. Sometimes we call the outside of the cell this extracellular matrix, right? It's just a mix of a bunch of fibers, right? You got some carbs and proteins and all kinds of other stuff hanging out here, right? Remember that lots of stuff is in the cell, but there's also lots of stuff in this fluid surrounding the cells. So let's go. We have what we call a fluid mosaic model to represent the cell membrane, and all that's going on around here. So let's break apart this phrase. Fluid, right? This is changing, it's moving. Mosaic, if you think of a mosaic, there's just lots of things put together, and this is a good model of how it works. So we have our cell membrane here, right? There's our membrane. That's our phospholipid bilayer. So if we're looking for phospholipids, there they are, right? Remember these little phospholipids? They're all down here, making up that membrane. There are also these embedded proteins, and we've talked about those already, right? They're embedded in the membrane. You will also find that another important structure in this is cholesterol, and I kind of drew over it, but you can see this molecule here. Cholesterol, right, it gets a bad name, but it's actually an important part of the membrane because it helps maintain this sort of fluid model. It helps the membrane to stay able to move and to be fluid. We also will find glycoproteins. So it's basically, if you break down the word, glycoproteins, kind of a sugary protein, right? So you see a protein here, it may have a sugar receptor or something on it. And then glycolipids as well. So in this case, you might see some lipid parts here that are anchored in the membrane, but it has some sugary things out here. So if you think about like what's going on, what are all these things? A lot of these are involved in transport and reception of things, right? Some things might need to come in, right? And they might be looking for the antenna. They might be looking for a receptor or something to attach to and come inside the cell. So there is a large mosaic model of everything going on around the membrane. Okay, so how do things move across? Well, let's think of a term called passive transport, right? If something happens passively, it doesn't require a lot of effort or energy. So passive transport is when things move from a high to a low concentration. So let's say you brought in some perfume into a room and you sprayed a bunch of it. When you bring it in, that perfume, those molecules are really highly concentrated in this bottle. Once you spray it into the air, those molecules can disperse throughout the room. So basically they're moving naturally from a high to a low concentration in the room. This doesn't require energy, right? The laws of the universe help dictate this. These molecules naturally want to move from a high to a low concentration, right? They're too crowded here. They want to move out some space. So this sets up diffusion and osmosis, two terms we want to talk about. Diffusion is just like you see right here in this example. So let's say we have a beaker. We have a semi-permeable membrane here in the center, and we got some molecules here. Let's say they're salt. If the membrane allows the salt to go across, right, it's big enough, it's got big enough holes that the salt can go through, or it has the right receptors, channel proteins for it to go through, the salt molecules, or sugar, whatever these molecules are, will naturally diffuse from a high to a low concentration. We call that diffusion. No energy is needed, and this is really important because when you got a high concentration of resources outside or inside the cell, or waste outside or inside the cell, they need to move across to where it's less concentrated. Diffusion, simple movement from a high concentration to a low concentration. Osmosis is basically just the diffusion of water. And my entire next video is on osmosis. We'll go into that into a lot greater detail. But the basics are water also moves from a high to low concentration. So over here, these particles, if they could move across a membrane, will move from high to low. But the water molecules themselves might also be moving from a high concentration of water to a low concentration of water. So take, for example, here. Let's say that we've got a U tube, and we take and we put, right now, it's pretty evenly distributed with, let's say this is some sugar, some glucose in there, right? The molecule is pretty evenly distributed. Let's say we added a bunch of sugar molecules to one side. Now, there's a higher concentration of sugar over here. So if there's a higher concentration of sugar molecules, that means there's a lower concentration of water now, right? So that means the water will want to move from over here, where there's a higher concentration of water, to there. So in this case, what you see is that the water will actually move in that direction. So because it will sort of in a way help to equalize or equilibrate from this side over here, where the ratio, right, of sucrose to water was more even as it was before, it will come over here. But the water is still moving from a high concentration of water to a lower concentration of water. So on this side now, right, because there was more sugar, there was thus a lower concentration of water, so it moved from a high concentration of water to a low concentration of water. You can see the direction of the water movement. Okay, so passive transport from a high concentration to a low concentration, and we can be talking about high concentration of molecules to a low concentration of molecules like sugar or salt. We often, though, are talking about movement of water, high concentration of water to a low concentration of water. And again, I have a whole video on that coming up. So let's do a little summary here. When things move easily across the membrane from a high concentration to a low concentration, we just call that simple diffusion. Okay, simple diffusion. High concentration of molecules to a lower concentration, they're going to move right across, assuming they can get across the membrane. Well, what if they can't get across, right? Maybe it's a charged particle, or maybe it's a larger molecule. Well, they're going to have to use a channel protein. We call this facilitated diffusion. We needed a channel protein to facilitate the movement of these molecules across. So notice we're still going from high concentration to a lower concentration. So these are no energy required, still going from high to low, diffusion and then facilitated diffusion. So occasionally, though, we need to go in the opposite direction. Let's say we want to go from a low concentration to a high concentration. And I'll give you some examples on the next slide of when we would do this. If we're in a low concentration and need to go to a higher concentration, this is called active transport, because we're actively going to have to use some ATP energy for this to occur. And we often call these pumps, right? In a way, they're pumping molecules across a membrane from a low to a high. So when we go from a low concentration to a high concentration, we're calling this active transport because it will need active use of energy, ATP energy. So I said I'd give you an example. Let's look at a specific example of when active transport is required. When we're going against the concentration gradient, right? So from low to high, we call these pumps often. They're made of proteins, and we're going to require some ATP energy. So the most famous pump, or I like to think the most famous pump, is the sodium-potassium pump in nerve cells, right? So when your neurons fire, they send information. What happens is we need to reset that. So we need to pump the sodium and potassium ions back so that the neuron can fire again. So this process requires energy, right? That's why your brain uses a lot of energy to run this pump. So here's a prime example. So this is the sodium-potassium pump here, and the neuron has fired. So the sodium has come inside, the potassium has gone outside, and we need to reverse this. We need to get the sodiums back out and the potassiums back in so that the nerve can fire again, or the neuron. So the first thing that happens is the sodium in the cytoplasm will bind to this pump. See them coming in. We'll use some ATP energy, right, to activate the pump. Notice that it is phosphorylated, right? So the ATP went to ADP and that little phosphate group went here. Activated the pump, and now it changed its conformation. So those molecules, right, that sodium can now freely float outside of the neuron, outside of the cell, and get ready to be used again. So then what about that potassium that was outside? So the potassium that was outside now will come, and notice it has to bind to a different spot in the sodium-potassium pump. And what you'll see is that when that happens, that phosphate group will be dephosphorylated, come off, opens back up, changing the conformation once again, going to the original conformation, and these potassium can release and go back inside the cell. So all we did here is move things against the concentration gradient, right? Because there was already a lot of sodium out here, but we needed to move more out. And inside, even though I don't show you a bunch, there was a lot of potassium inside. And so we needed to move the potassium back in to maintain that really high concentration gradient across the membrane. So this is a prime example of when we have to use energy to move things against their concentration gradient. One more example that I could tell you about is the idea that roots of a plant, this tree here for example, often have nitrate and phosphate pumps in their roots. Because if you think about it, the tree needs nitrogen, needs phosphate, needs nitrates and phosphates, but there's probably more in these roots and cells than in the soil, but we still need it, right? So we actually need that nitrogen, that phosphorus to come in the roots and be used by the plant, even if it's going against the concentration gradient. So it's very interesting that, of course, these are needed in the roots, so for the plant, but that the main, one of the main components of fertilizer, of course, is phosphorus and nitrogen, because that's what the plant needs to maintain its macromolecule level, to make all those important nucleic acids and proteins and things that are needed in the plant. Okay, so those are some examples of when we see active transport. The sodium-potassium pump in our nerve cells, and then also in plant cells in the roots, you'll see these phosphate and nitrate pumps as well. Okay, finally here, let's go back to where we started. Let's talk about when things need to come in the cell, or large molecules. Sometimes it's just best to do bulk movement. It's best to open up the garage door and just let everything come in, right? Instead of using that narrow front door. So bulk movement, when we need to move a lot, it happens through vesicles and vacuoles. Endocytosis, think endo, inside, is when molecules are taken into the cell from the plasma membrane. And so here we can see here, we've got some molecules, they need to come in, so a little bit of this plasma membrane will just pinch in, form a vesicle, move it inside, right? So we had a bulk movement that just needed to come inside the cell. So instead of messing with some sort of little protein or something over there, we're just going to bulk move them all in. That's called endocytosis, coming into the cell. The opposite, of course, of that would be exocytosis, when things need to exit. So in this case of exocytosis, we got some waste here. So it will move through a vesicle, bind to the membrane, and then this waste will be able to just be removed and pushed out of the cell. Exit exocytosis. So at the very beginning of the video, I said, what about these other things, large molecules, particularly viruses, right? What about SARS-CoV-2, the virus that causes COVID? Well, it actually needs to come into a cell. And these processes require energy. So, energy required for endo and exocytosis, and there's going to be some energy required for that particular virus to come in. How does it do it? Well, on the surface of the cells, I mentioned, are these receptors, right? So often, before endocytosis can occur, something's going to have to bind to that receptor, sort of like a key for the lock, so that the cell says, okay, receptor, mediate the entry into the cell, right? And there are tons of receptors because your cells need to move lots of materials around. There's a particular receptor called the ACE2 receptor involved in your body's angiotensin system, which basically allows things to move inside of the cell. Well, some viruses, in this case the coronavirus, actually hijacks these ACE2 receptors. This spike protein binds to the receptor, and then the cell says, okay, come on inside, endocytosis. It thinks it's something else. And then, of course, once that virus gets inside, it uses your host cell machinery to replicate, and then you've got a nasty infection. So it's crazy to think, right? All of this, all these diseases, this virus, all these different viruses that get into the cell are actually brought in via endocytosis because the membrane, which is there to protect us, does need to allow some things in. So something like a virus hijacks that system and pretends with this little spike protein to be something else. Your cells will naturally allow it to come in. So I want you to think about in this video all the ways things can move in and out of the cell, whether those things that are needed for the body or some foreign invaders that happen to take advantage of the plasma membrane and its systems.